Our quest to find life beyond Earth has accelerated dramatically in recent decades, driven by leaps in technology and an expanding understanding of the cosmos. Today, scientists employ a multifaceted approach that combines space missions, the search for biosignatures, and the study of exoplanets to uncover clues about life elsewhere. Each of these areas offers unique insights and challenges, and together, they pave the way for what might be the most profound discovery in human history. One of the most direct ways to find alien life is to examine environments within our own solar system where life as we know it might exist or might have existed in the past. Several current and planned space missions are focused on these promising locations. Mars has long been a candidate, given its past presence of water. NASA's Perseverance rover, which landed on Mars in 2021, is diligently collecting rock and soil samples from an ancient lakebed known as Jezero Crater. The goal is to search for signs of past microbial life trapped in the sediment and rock formations. Perseverance's onboard instruments analyze soil chemistry, search for organic molecules, and assess the planet's past habitability. Meanwhile, the icy moons of the outer solar system, such as Europa, Enceladus, and Titan, have become frontiers in the search for life. Underneath Europa's icy surface lies a global ocean of liquid water, warmed by tidal heating from Jupiter's gravity. NASA's upcoming Europa Clipper mission, set to launch in the mid-2020s, will fly multiple close passes to study the moon's surface and subsurface ocean, analyzing the ice and any plumes of water vapor that may erupt from cracks. Detecting the right chemical ingredients, such as salts, organic molecules, or even metabolic byproducts, could hint at an ocean environment where life might thrive. Enceladus, a small moon of Saturn, presented a surprising discovery when the Cassini spacecraft found plumes of water vapor and ice particles ejecting from its south polar region. These plumes contain organic compounds and suggest hydrothermal activity on the ocean floor, conditions that, on Earth, support thriving microbial ecosystems near deep-sea vents. While no dedicated mission to Enceladus is currently approved, concepts are under discussion to study these jets more closely or even capture samples for detailed chemical analysis. Titan, another moon of Saturn, offers a markedly different environment: a thick, nitrogen-rich atmosphere and hydrocarbon lakes of methane and ethane. Though very cold for Earth-like life, Titan's chemistry is intriguing and may support exotic life forms that use liquid methane as a solvent instead of water. The Dragonfly mission, launching in the mid-2030s, will send a rotorcraft lander to fly across Titan's surface, sampling organic compounds and assessing conditions that could support life. Beyond our solar system, the discovery and study of exoplanets, planets orbiting stars other than the Sun, has revolutionized the search for life. Since the first confirmed detection of an exoplanet in the 1990s, scientists have found thousands more using techniques such as the transit method, where a planet passes in front of its star and causes a slight dip in brightness, and the radial velocity method, which detects the wobble of a star caused by a planet's gravitational pull. Among these, a special focus is on planets located in the habitable zone around their stars, sometimes called the Goldilocks zone, where conditions might be just right for liquid water to exist on the planet's surface. While many discovered exoplanets are gas giants or otherwise inhospitable, a growing number of Earth-sized and super-Earth planets lie in their stars' habitable zones, raising tantalizing possibilities. One landmark discovery is the TRAPPIST T-1 system, consisting of 7 Earth-sized planets orbiting a dim, ultra-cool dwarf star about 40 light-years away. At least 3 of these fall within the habitable zone, where surface temperatures could allow liquid water. Their similar sizes to Earth make them prime targets for follow-up observations with powerful telescopes. Another notable target is Proxima Centauri b, the closest known exoplanet to Earth, orbiting Proxima Centauri, our nearest stellar neighbor. Although Proxima Centauri is a red dwarf star prone to stellar flares that might challenge an atmosphere's stability, the proximity of this planet makes it an intriguing subject for future studies. What scientists seek on these exoplanets are signs of life known as biosignatures, observable traits or substances that could indicate the presence of living organisms. Detecting biosignatures remotely is a major challenge and requires careful interpretation to avoid false positives. The most promising biosignatures are typically gases or chemical imbalances in a planet's atmosphere that would be unlikely to exist without biological activity. For instance, oxygen and ozone in Earth's atmosphere are closely linked to photosynthetic life. Methane is another gas of interest because it can be produced biologically, though it has geological sources as well. A simultaneous presence of oxygen and methane in an exoplanet's atmosphere could suggest ongoing biological processes, since these gases tend to react with each other and would need constant replenishment to coexist. The launch of the James Webb Space Telescope, JWST, in late 2021 has been pivotal in advancing these investigations. Unlike previous telescopes, JWST can observe in the infrared spectrum, which allows it to analyze the atmospheric composition of some exoplanets by measuring the light as it passes through or is emitted by their atmospheres. This capability gives researchers the chance to look for biosignature gases directly for the first time. Already, JWST has provided detailed spectra of exoplanet atmospheres, revealing the presence of water vapor, clouds, and other chemical components. Although detecting definitive signs of life remains elusive, these observations are setting the stage for increasingly sensitive surveys and refining our models for where life could be common or rare. Looking ahead, the field of astrobiology is preparing for new observatories and missions that will push the boundaries even further. Besides JWST, concepts such as the Large UV/Optical/Infrared Surveyor, LUVOIR, and the Habitable Exoplanet Observatory, HAB-X, envision telescopes capable of directly imaging Earth-like exoplanets and analyzing their atmospheres with unprecedented precision. Meanwhile, the Breakthrough Starshot initiative aims to send tiny, laser-driven probes to the Alpha Centauri star system within decades, allowing in situ examination of nearby exoplanets like Proxima b. Although still in the conceptual phase, such a mission represents a radical new approach to the search for life. The search for life is also deepening through the study of biosignatures closer to home. Scientists analyze meteorites for organic molecules and explore terrestrial analogs. Extreme environments on Earth, such as acidic lakes, deep subsurface rocks, or frozen deserts, that mimic conditions on other planets and moons. Understanding how life originates, survives, and leaves traces in these settings helps interpret the data we gather from other worlds. In parallel, the field is grappling with defining what exactly constitutes life and how to recognize it in unfamiliar contexts. Life, as we know it, is based on carbon chemistry and water, but scientists remain open to the possibility of alternative biochemistries, which complicates the search but also broadens our horizons. Together, these scientific advances and missions have brought us closer than ever to answering the age-old question: are we alone? While no definitive evidence of extraterrestrial life has yet emerged, the pace of discovery, both in our solar system and beyond, continues to accelerate. Each new finding refines our understanding of where life might exist and how we might recognize it, shaping a roadmap for the decades to come. As of now, the search for alien life remains a frontier marked by tantalizing hints, careful skepticism, and the hope that our methods will one day yield a positive detection. Whether that's the detection of microscopic organisms beneath a moon's icy crust, a subtle chemical signature in an exoplanet's atmosphere, or an entirely unexpected form of life, the efforts underway are steadily expanding the horizons of what human curiosity and ingenuity can achieve. The next decades will be critical in transforming our place in the cosmos, from a solitary observation to an informed participation in a broader tapestry of life. While the technological advances and scientific frameworks mentioned offer powerful tools for identifying life in the cosmos, they also highlight the complexity of the challenge. One emerging focus is the concept of biosignature gases beyond those typically associated with life on Earth. This broadening of parameters arises from the recognition that life elsewhere might exist under conditions very different from those on our own planet, potentially using alternative chemical processes or solvents. Earth-based life depends on liquid water as a solvent and carbon-based molecules for its structure. However, astrobiologists consider the possibility that life could arise using other solvents, such as liquid methane or ammonia. For instance, Titan's surface, host to vast lakes of methane and ethane, represents an environment where life, if it exists, may have evolved along these unconventional lines. Studying such environments forces scientists to rethink the signatures life might leave, including gases or chemical imbalances unlike those familiar in Earth's atmosphere. Beyond the search for specific chemicals, astrobiologists also try to understand the conditions necessary for life to originate and sustain itself, an area known as habitability research. Habitability depends on multiple factors, including the presence of liquid water, energy sources, and essential elements like carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, sometimes abbreviated as CHNOPS. Understanding these ingredients helps scientists narrow the list of candidate worlds where life might arise or survive. Habitability extends beyond the planet's surface. For example, subsurface oceans, shielded from harsh radiation and extreme temperatures, could provide stable environments for life. This idea underpins the interest in icy moons like Europa and Enceladus. Their subsurface oceans, protected beneath thick ice crusts, may bear closer resemblance to Earth's deep oceans around hydrothermal vents, where complex ecosystems thrive in total darkness. In recent years, the study of extremophiles, organisms capable of surviving and thriving under conditions once thought inhospitable, has reshaped our perspective on where life might exist. On Earth, microbes have been found living inside rocks miles beneath the surface, in acidic hot springs, in high radiation zones, and in Antarctica's subglacial lakes. These discoveries reveal that life is remarkably adaptable, expanding the range of environmental conditions deemed potentially habitable. For example, bacteria found near hydrothermal vents on Earth's ocean floor utilize chemical energy from minerals emitted by the vents, rather than relying on sunlight. The discovery of such chemosynthetic ecosystems has significant implications for extraterrestrial life, as it suggests that planets or moons without sunlight but with internal geochemical activity might still harbor life. That insight directly influences missions like the proposed Ice Clipper and other concepts designed to sample the plumes of Enceladus or to drill through Europa's ice shell, searching for signs of microbial life or the chemical foundations needed to support it. If these missions detect complex organic molecules or metabolic byproducts, they would constitute strong evidence that life, or at least prebiotic chemistry, exists beneath these icy surfaces. Additionally, the study of atmospheric biosignatures is progressing in sophistication. Beyond oxygen and methane, scientists are identifying other potential biosignature gases that, when detected in combination or in unexpected amounts, could signal biological processes. For example, phosphine pH3, a gas produced by some anaerobic microorganisms on Earth, garnered attention when tentative detections were reported in the clouds of Venus. Although initial observations and interpretations remain contentious, this underscored how new molecules might challenge existing assumptions and prompt a reevaluation of where to look for life. Another aspect to consider is the concept of false positives and false negatives in life detection. False positives occur when non-biological processes mimic signatures typically attributed to life, such as certain chemical reactions driven by geological phenomena. False negatives happen when life is present, but its signatures are too subtle or masked by environmental factors to be detected. Scientists emphasize the necessity of corroborating multiple lines of evidence before concluding that life has been found to avoid premature claims. Interdisciplinary collaboration is crucial in this field. Geologists, chemists, biologists, planetary scientists, and astronomers work together to interpret data accurately. For example, understanding a planet's geological cycles helps inform which gases might accumulate or be depleted in the atmosphere naturally versus biologically. Likewise, detailed models of atmospheric chemistry help separate biological signals from abiotic background noise. Alongside observational missions, laboratory research and computer simulations contribute to the search for life. Laboratory experiments simulate extraterrestrial conditions, extreme cold, high radiation, low pressure, to observe how organic molecules might form, persist, or interact. Some experiments probe the plausibility of alternative biochemistries, like life based on silicon or solvents other than water, though none have yet produced clear evidence of viable alternative forms. Another promising avenue is the future development of instruments capable of detecting complex organic molecules and potential biosignatures with greater sensitivity and specificity. Mass spectrometers, gas chromatographs, and advanced spectroscopic techniques are evolving to scan samples from planetary surfaces, atmospheres, and cometary or asteroid material. Such instruments are integral to planned missions like the Mars Sample Return, which could bring pristine Martian rocks to Earth laboratories for detailed analysis. In the realm of exoplanet research, the tiny signals of potential biosignatures require enormous technological leaps and patience. The atmospheres of these distant worlds are incredibly faint and often obscured by clouds, hazes, or their stars' overwhelming brightness. Innovative techniques, including high-contrast imaging and starshades, spaceborne devices designed to block out starlight, seek to improve our ability to directly image Earth-like planets and analyze their atmospheres. Observing exoplanets in multiple wavelengths, from ultraviolet to infrared, allows scientists to profile a range of molecules and environmental factors. Combining atmospheric data with measurements of planetary mass, radius, and orbital characteristics helps reconstruct a world's overall habitability. Studying a planet's climate dynamics and response to stellar activity adds further context essential to assessing its potential to support life. Importantly, the search for extraterrestrial life also prompts reflection on the philosophical and societal dimensions of such a discovery. Finding even microbial life elsewhere would profoundly impact our understanding of biology, evolution, and our place in the universe. It would raise ethical questions about planetary protection, the preservation of alien ecosystems, and the responsibilities of explorers and researchers. To prepare for these implications, organizations like the United Nations and various space agencies have developed frameworks aimed at preventing the contamination of other worlds by Earth life, and vice versa. This planetary protection ensures that future missions do not compromise sites of possible biological interest, preserving scientific integrity and honoring potential alien life forms. Considering the scale of cosmic distances and the complexities of life detection, many experts emphasize the importance of patience and persistent effort. The journey to find life beyond Earth is a marathon, not a sprint, involving incremental advances and the continual refinement of hypotheses. Each mission, observation, and experiment contributes a piece to the puzzle, gradually enhancing our capacity to answer one of humanity's oldest questions. Though definitive evidence of extraterrestrial life has yet to be found, the increasing number of potentially habitable environments, both within our solar system and among exoplanets, combined with rapidly advancing technologies, make the prospect of discovery more realistic than ever. As we expand our reach and sharpen our tools, the search is not just about finding alien life, but about uncovering the processes and conditions that make life possible, enriching our appreciation of the natural universe. Ultimately, the question, how close are we to finding alien life, eludes a simple answer. It depends on factors both within and beyond our control: technological capabilities, scientific understanding, the true diversity of life in the universe, and opportunities presented by cosmic events or planetary conditions. Yet the narrative unfolding shows steady progress, marked by human ingenuity, and an enduring spirit of inquiry that propels the quest onward. As we explore the search for alien life, an important aspect to consider is the role of planetary environments themselves, how they form, evolve, and interact with their stars and surroundings. Understanding planetary atmospheres and surfaces is crucial because these factors strongly influence a world's habitability and the likelihood that life could develop or endure there. Within our solar system, the diversity among planets and moons illustrates the range of potential habitats. Mars, for example, has long been a focal point in astrobiology due to evidence that it once had flowing water on its surface, rivers, lakes, and possibly even shallow seas. Today, Mars is cold and dry, with a thin atmosphere mostly of carbon dioxide, but it still offers clues that could reveal past or present microbial life. Subsurface ice deposits and recurring slope lineae, dark streaks that appear seasonally and may indicate briny liquid flows, fuel ongoing debates about where life might exist beneath or just below the Martian surface. Future missions, such as the rover Perseverance and planned landers, focus on collecting and analyzing rock and soil samples that might preserve ancient organic compounds or microfossils. These samples could help clarify whether life ever arose on Mars and provide valuable benchmarks for detecting biosignatures elsewhere. Venus presents a starkly different environment. Its surface is scorching hot, around 460 degrees Celsius, with crushing pressure and clouds of sulfuric acid. This extreme environment appears inhospitable to life as we know it. However, the intriguing possibility of life existing high in the Venusian cloud layers, where temperatures and pressures are less extreme, has prompted renewed scientific interest. Certain atmospheric anomalies, such as mysterious absorption features and the controversial detection of phosphine gas, hint at chemical processes that are not yet fully understood and may, or may not, be linked to living organisms. Beyond our solar system, the discovery of exoplanets over the past few decades has revolutionized the scope of astrobiology. Thousands of these worlds orbit other stars, displaying a vast variety of sizes, compositions, and orbital arrangements. Among them, the biggest prize is Earth-like planets located within the habitable zone, the region around a star where temperatures would allow liquid water to exist on a planet's surface. One prominent example is the TRAPPIST T-1 system, which contains 7 Earth-sized planets orbiting an ultra-cool dwarf star about 40 light-years away. 3 of these planets lie within the habitable zone, offering promising candidates to investigate whether conditions suitable for life exist there. However, habitability depends on much more than just distance from the star. Factors such as the planet's atmosphere, magnetic field, geologic activity, and water inventory all influence if life can be sustained. Characterizing exoplanet atmospheres remains a demanding technical challenge. Current space telescopes like Hubble and the recently launched James Webb Space Telescope, JWST, have the capability to probe the atmospheres of select exoplanets by observing how starlight filters through them during transits, when the planet passes in front of its star. These observations can detect the presence of gases like water vapor, carbon dioxide, methane, and even oxygen, each potentially serving as a hint toward biological activity. One aspect that complicates the interpretation is the fact that many abiotic, non-living processes can produce these gases. For instance, methane can be released through volcanic activity or chemical reactions unrelated to life. Similarly, oxygen can accumulate from water vapor being split by ultraviolet light, a process called photodissociation, without any life needing to be involved. Therefore, identifying combinations or ratios of gases that are unlikely to arise without biology becomes vital to avoid false positives. Another frontier in the search for alien life is the study of so-called technosignatures, evidence of advanced civilizations capable of creating detectable alterations in their environment. These might include unusual electromagnetic signals, like radio waves, large-scale construction projects, such as Dyson spheres, hypothetical megastructures built around stars to harness energy, or even industrial pollution in planetary atmospheres. So far, no confirmed technosignatures have been detected, but ongoing efforts like the Search for Extraterrestrial Intelligence, SETI, continue to scan the skies across multiple wavelengths for intentional or accidental signals. Technosignatures broaden the scope of life detection beyond primitive microbes to include the possibility of intelligent life. However, this search is challenged by the vast volumes of space and time over which signals might disperse or remain detectable. The silence so far could reflect the rarity of technological civilizations, the short temporal window during which signals are emitted, or the limitations of our current observational methods. In considering how close we are to finding alien life, it helps to reflect on the scale of time and space involved. The universe is approximately 13.8 billion years old, while Earth's history has seen life for around 3.5 billion years. Life here began relatively soon after the planet cooled enough to host liquid water, suggesting that under the right conditions, life may arise quickly. On the other hand, complex multicellular life took billions of years to evolve, and only recently on a cosmic timescale has intelligent life emerged. If these patterns hold elsewhere, microbial life might be quite common in the universe, even if complex or technological life is far rarer. Arguments like this are central to the famous Drake equation, a probabilistic framework formulated to estimate how many civilizations capable of communication might exist within our galaxy. While many parameters of the equation remain uncertain, it helps guide researchers to where discoveries might be most fruitful and what kinds of data would refine the estimates. Back on Earth, the study of life's chemical origins, called abiogenesis, also provides important context. Understanding how life emerged from non-living chemistry lays groundwork for assessing how likely life might appear on other worlds. While the exact path remains unclear, experiments have demonstrated that organic molecules, amino acids, nucleotides, simple sugars, can form spontaneously under plausible early planetary conditions. The challenge lies in bridging the gap from these molecules to self-replicating, metabolizing cells. Exploring possible pathways to abiogenesis on other worlds helps diversify what we look for in the cosmos. For example, instead of Earth-like cells based on DNA and proteins, life elsewhere might use alternative biochemistry, depending on local geology and chemistry. This realization has encouraged scientists to look for organic chemistry signatures in diverse environments, not just those that mimic Earth's. Looking further ahead, upcoming missions and observatories promise to expand the frontiers of our search. NASA's Europa Clipper, set for launch in the mid-2020s, will conduct multiple flybys of Jupiter's icy moon Europa, probing its subsurface ocean and measuring surface chemistry. ESA's JUICE, Jupiter Icy Moons Explorer, will explore Europa alongside Ganymede and Callisto, investigating their habitability potential. Additionally, proposed concepts like LUVOIR, Large UV/Optical/IR Surveyor, and HAB-X, Habitable Exoplanet Observatory, are large space telescopes envisioned to directly image Earth-like exoplanets and analyze their atmospheres. By combining ultra-high resolution, sensitivity, and advanced technologies like starshades to block out starlight, these observatories aim to detect biosignatures and possibly telltale signs of life in great detail. Despite the exciting prospects, a key part of progress lies in collaboration and the integration of diverse methods. The conjunction of theoretical modeling, laboratory simulations, robotic exploration, space telescopes, and observational astronomy creates an increasingly powerful approach to detect and confirm life beyond Earth. Still, caution remains essential. History has witnessed moments when hopeful claims of extraterrestrial life were later shown to have alternative explanations. Maintaining rigorous scientific standards, transparency, and critical analysis ensures that any future discovery withstands intense scrutiny, solidifying its place in knowledge. One of the most significant issues is how to distinguish the true signals of life from noise. For example, the Viking missions to Mars in the 1970s conducted experiments to detect metabolism in Martian soil. Initial results were ambiguous, with some suggesting microbial activity, but others explained by non-biological chemical reactions. This experience underscored the difficulty of unambiguous life detection and the need for multiple complementary approaches. In the search for life signatures, scientists often emphasize the combined presence of several indicators rather than reliance on a single measurement. For instance, the simultaneous detection of oxygen and methane, both gases that can react with each other rapidly, might suggest continuous replenishment by living organisms. Finding such a disequilibrium state in an exoplanet's atmosphere would be compelling evidence of life, warranting follow-up investigation. Moreover, detecting life inside planets covered by thick ice or deep oceans demands innovative technology. The presence of ice shell tens of kilometers thick on moons like Europa presents a formidable barrier. Drilling through such ice or accessing ocean plumes ejected by cryovolcanism requires carefully designed instruments, robust engineering, and significant investment. These challenges illustrate that while we have compelling targets, reaching and sampling them is no small feat. To summarize where we stand, we have an expanding roster of likely habitats, advancing methods to observe and analyze them, and a richer understanding of life's versatility. But the direct detection of alien life remains just beyond current capabilities. The progress is steady and often incremental rather than revolutionary. Thus, the search for extraterrestrial life is both a scientific endeavor and a grand exploration that transcends disciplines and cultures. It taps into humanity's curiosity about ourselves and the universe. While we don't yet have definitive answers, each step taken brings us closer to what might be one of the most profound discoveries in history. In the meantime, the challenge continues to inspire technological innovation, international cooperation, and the honing of scientific methods. Whether through the next rover sent to Mars, observations of distant exoplanet atmospheres, or the drilling into icy moons, humanity is steadily advancing toward answering the question: how close are we to finding alien life? The answer is complex, close enough to be hopeful, distant enough to remain patient, and in every way a testament to our yearning to know if we are alone. Another vital dimension to the quest for alien life involves the study of extremophiles here on Earth, organisms that thrive in conditions once thought too harsh for life. These resilient forms of life expand our understanding of where and how biology can exist, effectively broadening the range of environments considered potentially habitable beyond the classic, Earth-like template. Extremophiles inhabit places characterized by extreme temperature, acidity, salinity, radiation, pressure, or nutrient scarcity. For example, thermophiles flourish in the near-boiling waters of deep-sea hydrothermal vents, where superheated, mineral-rich water gushes from the ocean floor. These vents feature complete darkness and crushing pressures thousands of meters beneath the surface. Yet microbial communities not only survive, they flourish. In such ecosystems, organisms depend on chemosynthesis rather than photosynthesis, using chemical energy from sulfur or methane compounds to build organic molecules. This discovery reshaped astrobiologists' expectations by showing that life does not require sunlight and can adapt to entirely different energy sources. Similarly, acidophils thrive in highly acidic environments like the Rio Tinto River in Spain, where the water's pH can be as low as 2, akin to stomach acid. Organisms endure and metabolize in these seemingly inhospitable waters, revealing how life can adapt to chemical extremes. Likewise, halophiles inhabit salt-saturated lakes and even salt crystal-filled caves, drawing energy and nutrients in conditions that would dehydrate most other cells. These findings have profound implications for our search beyond Earth. For instance, many moons and planets with plentiful water may also have environments where life relies on chemical energy rather than sunlight. Europa's subsurface ocean, kept liquid beneath a thick icy shell by tidal heating, is a prime candidate for life sustained by hydrothermal vents on its ocean floor, analogous to those on Earth. Enceladus, another icy moon of Saturn, actively ejects plumes of water vapor and organic compounds through fissures in its ice, hinting at subsurface hydrothermal activity. Missions like NASA's planned Europa Clipper aim to analyze these plumes for amino acids, salts, and other chemicals associated with life on Earth. Beyond our solar system, extremophile adaptation informs how we interpret exoplanet environments. For example, planets orbiting red dwarf stars often receive fluctuating stellar radiation and may be tidally locked, with one hemisphere in perpetual daylight and the other in unending night. Such conditions differ dramatically from Earth's, but the presence of extremophiles on Earth suggests that life could persist by exploiting niches where temperature, radiation, or chemical gradients allow survival in an alien landscape. Studying extremophiles also affects the design of life detection instruments. Knowing that some microbes metabolize sulfur compounds or subsist in near-zero oxygen conditions leads to targeted searches for specific biomolecules or chemical reactions. Future missions might incorporate sensors capable of detecting unusual redox, oxidation reduction, states indicative of life processes distinct from terrestrial norms. These efforts emphasize that life as we do not know it can only really be meaningfully explored after we thoroughly understand the many ways life on our own planet has adapted and diversified. Aside from microbial life, an ongoing question is the likelihood of more complex or intelligent life beyond Earth. On our planet, complex multicellularity only appeared after billions of years of relatively simple life, and intelligence capable of technological achievements is a recent development by comparison. The apparent absence of detected extraterrestrial civilizations, despite decades of searching, has been encapsulated in what is known as the Fermi paradox. If intelligent life is common, why haven't we observed any signs of it? Scientists approach this paradox by considering multiple explanations, ranging from practical limitations in our detection methods to the possibility that technological civilizations tend to self-destruct or are rare by cosmic standards. One idea is that the Great Filter, a series of difficult evolutionary or societal hurdles, might drastically reduce the prevalence of advanced civilizations. The nature of this filter remains speculative, but could involve challenges such as the origin of life itself, the jump to intelligence, or the sustainability of civilization. While this debate is ongoing, research into astrobiology focuses on gathering empirical evidence to constrain possible answers. For example, searches for so-called technosignatures aim to detect signals or artifacts indicative of intelligent activity. These include radio and optical signals, laser pulses, or irregularities in stellar brightness, caused by massive engineered structures. SETI initiatives have expanded in recent years, scanning broader ranges of frequencies and enhancing sensitivity with improved technology. But no definitive artificial signals have yet been found. In addition to electromagnetic signals, scientists also consider chemical signatures of industrial activity, such as synthetic pollutants in exoplanet atmospheres. Earth's own atmosphere has dramatically changed due to anthropogenic activity. The buildup of chlorofluorocarbons, CFCs, nitrogen oxides, and other compounds could, in principle, be detected from great distances if similar processes occurred elsewhere. Identifying an atmospheric composition inconsistent with natural processes might provide an indirect clue to the presence of intelligent life. Despite the lack of confirmed technosignatures, these efforts emphasize a key point. Detecting life on the scale of civilizations is far more challenging than identifying microbial life. Civilizations may be short-lived in cosmic terms, signals weaken and disperse over vast distances, or advanced beings might communicate or exist in ways unfamiliar to humanity. This deep uncertainty encourages continued broad-ranging strategies that include both biosignature and technosignature searches. Turning back toward biological signatures, laboratory analysis of meteorites and interplanetary dust particles offers another window into the prevalence of life's building blocks. Carbonaceous chondrites, a class of meteorites originating from asteroids, contain complex organic molecules including amino acids and nucleobases, the components of proteins and DNA/RNA. These discoveries suggest that the raw ingredients for life are widespread in the solar system and potentially throughout our galaxy. One famous example is the Murchison meteorite, which fell in Australia in 1969 and was extensively studied for its rich organic content. Some of the amino acids found in it are not common on Earth and exhibit unusual isotopic ratios that indicate an extraterrestrial origin. Such findings support the idea that organic chemistry is not unique to Earth, but is naturally generated and delivered by cosmic processes like interstellar dust formation and asteroid collisions. Additionally, recent analyses of cometary material from missions like Rosetta's study of Comet P/Churyumov-Gerasimenko reveal abundant organic compounds, including complex hydrocarbons and oxygen-bearing molecules. Comets and asteroids are thought to have contributed not only water, but also prebiotic organics to early Earth via impacts, potentially aiding the emergence of life. These insights open the possibility that life, or at least its molecular precursors, might be seeded throughout planetary systems, a hypothesis known as panspermia. While this remains hypothetical and does not explain how life originated initially, it provides a framework for understanding how life might spread and suggests broader zones of habitability across star systems. Advances in laboratory simulations continue to improve our understanding of prebiotic chemistry. Scientists recreate conditions thought to resemble early Earth or other planetary environments, varying temperature, pressure, radiation, and chemical composition, to observe the spontaneous formation of complex organic molecules and even protocells. These studies underscore how molecules crucial for life can assemble in diverse contexts, providing experimental support for astrobiological theories. Another critical aspect is the use of machine learning and artificial intelligence, AI, to sift through the enormous volume of data collected by telescopes, spacecraft, and laboratory experiments. Automated pattern recognition helps identify potential biosignatures or technosignatures more efficiently than manual analysis. As the quantity of observed exoplanets and cosmic phenomena grows exponentially, AI will become an indispensable tool to guide researchers toward the most promising candidates for follow-up study. Collaboration between multiple scientific disciplines, astronomy, geology, chemistry, biology, planetary science, is increasingly essential. For example, Understanding an exoplanet's interior composition or geological activity requires comparative planetology, drawing from observations of Earth and other solar system bodies. Predicting atmospheric chemistry depends on detailed modeling of photochemical reactions influenced by stellar radiation. Detecting life, therefore, isn't just about looking for one signature, but assembling a coherent picture from diverse clues. International cooperation also plays a role, especially in funding and coordinating ambitious projects like space telescopes and planetary missions. With the inherent challenges and costs of exploring beyond our planet, pooling expertise and resources accelerates progress and enables more comprehensive investigations. In contemplating the future, planned and proposed projects promise significant leaps. The James Webb Space Telescope, operational since late 2021, has already demonstrated the ability to analyze planetary atmospheres with unprecedented sensitivity. Looking further ahead, telescopes like the Extremely Large Telescope, ELT, on the ground, and concepts such as the Origin Space Telescope aim to access a wider range of wavelengths and improve resolution to reveal finer atmospheric details. Moreover, missions targeting icy moons, including NASA's Dragonfly mission to Titan and further planned landers to Europa and Enceladus, will explore rich chemical environments closer to home. These efforts bring the promise of in situ analysis of potential habitats and may provide the first direct evidence for life beyond Earth. The search also encompasses the emerging field of astrogeochemistry, the study of how geological and biological processes intertwine. Understanding the cycling of elements like carbon, sulfur, and phosphorus within planetary systems offers critical context to identify unusual patterns that might indicate life's influence. Finally, the philosophical and societal implications of discovering extraterrestrial life guide much of astrobiological inquiry and public engagement. Finding even microbial life elsewhere would transform our understanding of life's universality and the uniqueness of Earth. Detecting intelligence would prompt profound questions about civilization, communication, and humanity's place in the cosmos. In the current era, with steady technological progress and expanding knowledge, the search for alien life is more focused and informed than ever. While definitive evidence remains elusive, the ongoing accumulation of data, enriched by cross-disciplinary insights and cutting-edge technology, brings us closer to answering one of humanity's oldest questions: are we alone in the universe? The answer, when it comes, promises to revolutionize science and culture alike, offering a new perspective on life, both terrestrial and cosmic. As the search for life continues, one particularly powerful avenue lies in the study of exoplanets, planets orbiting stars beyond our solar system. Since the first confirmed detection of an exoplanet orbiting a sun-like star in 1995, astronomers have discovered thousands of such worlds using various methods. Each new discovery helps paint a more detailed picture of the diversity of planetary systems and, critically, the frequency of worlds that might support life. The most prolific technique for exoplanet detection has been the transit method, where a planet passes in front of its host star relative to our line of sight, causing a slight, periodic dimming of the star's brightness. This technique not only reveals the presence of a planet, but can also offer clues about its size, orbital distance, and even atmospheric properties. NASA's Kepler Space Telescope revolutionized this field by discovering over 26 confirmed exoplanets, many located within the so-called habitable zone, the region around a star where liquid water could exist on a planet's surface. However, the habitable zone is a simplified concept. Habitable conditions depend on many factors beyond distance from the star, including the planet's atmosphere, geological activity, magnetic field, and stellar radiation environment. For example, Venus lies within the Sun's habitable zone, but has an inhospitable, greenhouse gas-dense atmosphere, leading to surface temperatures high enough to melt lead. Conversely, Europa, A moon of Jupiter far outside the solar system's habitable zone harbors a subsurface ocean kept warm by tidal heating, showcasing how diverse pathways to habitability can be. Characterizing exoplanet atmospheres has become a critical focus, particularly in the quest for biosignatures or indicators of life. When a planet transits its star, starlight filters through its atmosphere, imprinting spectral signatures of gases present. By analyzing these spectra, scientists can detect molecules like water vapor, carbon dioxide, methane, and oxygen. Some combinations of these gases might indicate biological activity, especially if out of chemical equilibrium, such as oxygen and methane coexisting in significant amounts, since these molecules tend to react with each other and therefore require continual replenishment. The James Webb Space Telescope, JWST, has already begun probing exoplanet atmospheres with remarkable sensitivity, targeting smaller, rocky planets rather than just large gas giants. Its findings could identify potential signs of habitability or life, but interpreting these signals demands caution. Non-biological processes can produce many of the same gases, and understanding a planet's geological and atmospheric context is crucial to avoid false positives. Another emerging technique, direct imaging, aims to capture actual pictures of exoplanets by blocking the overwhelming glare of their host stars. This approach allows detailed observations of a planet's reflected light, which carries information about surface properties and atmospheric composition. Although challenging due to tiny planet-to-star contrasts and immense distances, advances in coronagraphs and starshades, devices designed to suppress starlight, are pushing the frontiers of this method, promising breakthroughs in the coming decades. Meanwhile, the search for biosignatures is not limited to atmospheric gases. Scientists also contemplate surface features that might indicate life, such as the red edge effect caused by vegetation reflecting near-infrared light, or seasonal changes hinting at biological activity. While these signals would be subtle and difficult to detect at interstellar distances, future telescopes with higher resolution and sensitivity may unlock this potential. Looking inward within our solar system, detailed explorations of Mars continue to be a priority. Past missions have discovered compelling evidence that Mars once possessed flowing water, lakes, and perhaps even oceans. The planet's ancient surface shows minerals like clays and sulfates associated with watery environments, suggesting that conditions were once more favorable for life. Moreover, methane detections in Mars' atmosphere, though still debated, raise intriguing questions, since methane on Earth is commonly produced by biological processes. Current missions, such as NASA's Perseverance rover, are equipped to search for signs of past life by collecting rock samples and analyzing their chemical and isotopic properties. The rover's hunt focuses on identifying organic compounds and microfossil-like structures trapped in sedimentary rocks. These carefully curated samples could one day be brought back to Earth for detailed laboratory study, offering the most direct evidence of past Martian life. Further afield, icy moons like Europa and Enceladus remain among the most promising places to find extant life or convincing biosignatures. The discovery of plumes ejecting water from Enceladus' subsurface ocean and the possibility of hydrothermal vents on its ocean floor raise the possibility of habitats analogous to those supporting life near Earth's deep-sea vents. Although no mission has directly landed on these moons' oceans yet, future projects may deploy sophisticated probes to penetrate the ice and analyze water chemistry, perhaps even capture microorganisms if found. Titan, Saturn's largest moon, presents a unique case where life might exist in a very different chemical environment. Titan's thick nitrogen atmosphere and surface lakes of liquid methane and ethane create an exotic setting where life, if present, might be based on non-water solvents. Studying such possibilities challenges the Earth-centric assumptions about biochemistry and broadens the conceptual framework of habitability. While our probes steadily approach these intriguing worlds, the search for life is also spurred by theoretical work in understanding life's origins and limits. Researchers employ chemical and biological models to investigate how primitive molecules might evolve into self-replicating systems. Experiments in the lab simulate conditions of early planets or moons, revealing how amino acids, nucleotides, and lipid membranes can form spontaneously under certain scenarios. Understanding these pathways helps constrain where life could emerge and what signs it might leave behind. One growing area is the study of chirality, the handedness of biological molecules. Life on Earth exclusively uses left-handed amino acids and right-handed sugars. Detecting a preference for one chirality over another in organic molecules found on other worlds could serve as a strong biosignature, since abiotic processes generally produce racemic mixtures, equal amounts of left and right-handed forms. Analytical techniques under development aim to detect and measure molecular chirality remotely or in situ. Another key consideration is the temporal dimension of life. Planets or moons may undergo cycles where conditions pass intermittently into and out of habitability. Transient environments, such as melting ice caps or volcanic activity, may provide only brief windows for life to emerge or be detectable. This dynamic aspect complicates the search and suggests timing missions and observations matters greatly. Despite the many promising leads, we must also acknowledge the formidable challenges. One is the vastness of space and the scarcity of direct contact opportunities. Even with thousands of exoplanets detected, the sheer distance, often tens to hundreds of light years, means that sending probes or receiving detailed data takes enormous time and resources. For now, much discovery will come from telescopic observation and robotic missions within our solar system. Furthermore, distinguishing unambiguous biosignatures remains a fundamental hurdle. Planetary environments are complex, and many processes can mimic biological signals. For example, volcanic activity can release methane, and photochemical reactions in atmospheres can produce oxygen or ozone without life. Interpreting such signatures demands comprehensive understanding of planetary context and sophisticated models to rule out false positives. The interplay between life and environment also raises intriguing possibilities of feedback systems that may alter planetary atmospheres or surfaces over time. Earth's own oxygen atmosphere is a product of billions of years of photosynthesis and significantly modified by life. Detecting similar biosphere-driven changes elsewhere would be compelling, but requires observing planets across a range of ages and evolutionary stages. Looking ahead, interdisciplinary collaboration and technological innovation will be pivotal. Next-generation telescopes, such as the planned LUVOIR, Large UV Optical Infrared Surveyor, and HAB-X, Habitable Exoplanet Observatory, space telescopes aim to characterize Earth-like exoplanets with even greater detail. On the ground, extremely large telescopes equipped with advanced adaptive optics and spectrometers will enable unprecedented precision in studying nearby planetary systems. In tandem, missions exploring ocean worlds and sample returns from Mars promise in situ data crucial for identifying biological signatures beyond ambiguous remote observations. Advances in miniaturized instruments, autonomous systems, and contamination prevention will enhance these probes' ability to detect life if it exists. Public engagement and philosophical reflection remain important components. The discovery of extraterrestrial life, even at the microbial level, would reshape countless disciplines, from biology and chemistry to sociology and theology. Preparing for such an event encourages thoughtful discussions on ethics, planetary protection, and the impact on human society. Currently, the search for alien life sits at a transition point, from the purely theoretical and distant to direct investigation enabled by technological breakthroughs. While definitive evidence remains to be found, the broad expanse of knowledge gained and tools developed nurtures growing optimism. Each new discovery, whether a habitable exoplanet, an organic-rich meteorite, or a plume from an icy moon, adds a piece to the cosmic puzzle. In sum, humanity's exploration for life beyond Earth is progressing steadily, but cautiously, guided by both curiosity and rigorous science. The challenges are immense, but the scientific community is equipped with an ever-expanding toolkit and expanding conceptual frameworks. Whether life elsewhere is abundant or rare, simple or complex, the continued quest to find out enriches our understanding of ourselves and our place in the universe. Another critical piece of the puzzle in our quest to find alien life is the study of astrobiology's biggest question. How did life originate on Earth in the first place? Understanding life's beginnings here provides a template for what to look for elsewhere and how to recognize life's earliest signs. This challenge is immense because life's origins date back roughly 4 billion years, a period from which few direct clues remain. However, scientists have made significant strides by examining Earth's oldest rocks, simulating ancient environments in laboratories, and studying extreme environments where life perseveres today. One of the leading hypotheses is that life began in hydrothermal vent systems on the ocean floor. These vents spew mineral-rich, warm water, creating energy gradients and chemical environments favorable for the emergence of life's building blocks. Laboratory experiments have demonstrated that amino acids and nucleotides, the basic components of proteins, and RNA/DNA, can form spontaneously under such conditions. Furthermore, these vents provide a protected environment against harsh surface conditions, such as intense ultraviolet radiation and meteor impacts that characterized Earth's early history. Another promising avenue is the RNA world hypothesis, which suggests that self-replicating RNA molecules preceded DNA and proteins in early life forms. RNA can both store genetic information and catalyze chemical reactions, potentially bootstrapping the complexity required for living systems. Researchers have successfully synthesized RNA-like molecules and observed their ability to evolve within controlled environments, lending support to this idea. If RNA or similar molecules are detected on other worlds, it could be a strong indicator of early or primitive life. But the origin of life is not just a matter of chemistry. It also involves the emergence of cellular structures that can enclose and protect biochemical reactions. The spontaneous formation of lipid membranes in primordial conditions is another significant finding, showing how primitive protocells could have arisen. These membranes create compartments essential for life's processes, helping molecules concentrate and interact more efficiently. By piecing together these steps, from simple molecules to self-replicating entities enclosed in membranes, scientists establish criteria for detecting signs of life's early stages outside Earth. For example, detecting complex organic molecules combined with evidence of energy flows and compartmentalization could suggest life or pre-life chemistry. Missions to ice-covered moons or sample returns from Mars could test these ideas by searching for organic chemistry signatures matching those predicted by prebiotic models. Life's origins also connect closely with planetary environments and their evolution. Conditions on a planet or moon must be sustained long enough to allow this complex chemistry to develop and transition into living organisms. On Earth, the emergence of life is thought to have happened relatively quickly after the planet cooled sufficiently to support stable oceans, roughly 4 billion years ago. This rapid appearance suggests that life might arise readily given the right conditions, although this remains an open question. The concept of habitability, therefore, extends into time as well as space. A planet's habitability might be transient, influenced by factors like volcanic activity, impact events, magnetic field strength, and atmospheric evolution. For instance, Mars likely had habitable conditions early on but lost its atmosphere and water over time, shrinking the window for life. In contrast, Earth has maintained stable conditions for billions of years, enabling complex life to evolve. This temporal dimension raises expectations for the types of life we might find. In some worlds, life might be ancient and well-established, potentially evolving complex forms or even intelligence. In others, particularly those with intermittent or evolving conditions, life might be microbial, chemotrophic, or dormant, waiting for favorable conditions to become active. Recognizing these possibilities requires flexibility in both detection strategies and definitions of life. Detecting such life hinges on increasingly sophisticated instruments and analysis techniques. Modern spectrometers can identify trace gases and isotopic ratios that hint at biological processes. Isotopic signatures are especially telling because living organisms tend to favor lighter isotopes, a process that can leave characteristic patterns in rocks and atmosphere. For example, the ratio of carbon-12 to carbon-13 isotopes in organic matter differs significantly from inorganic carbon sources, making isotope analysis a powerful tool in identifying biological activity. Similarly, the detection of complex organic molecules, such as polycyclic aromatic hydrocarbons, PAS, amino acids, or even simpler biomolecules, in meteorites and comets suggests that the precursors to life are widespread in the cosmos. The Murchison meteorite, which fell in Australia in 1969, contained over 70 different amino acids, some not found on Earth. These findings imply that the chemical inventory needed for life's origins may be common throughout the galaxy, delivered through cosmic dust and impacts. Complementary to this chemical approach is the study of extremophiles, organisms capable of surviving in conditions once thought inhospitable for life. Discoveries of microbes thriving in boiling hot springs, acidic lakes, deep subsurface rocks, and highly radioactive environments have significantly expanded our understanding of life's resilience and adaptability. This knowledge informs us not to limit the search based only on Earth-like conditions, but to consider a broader range of environments where life might persist. An exciting development in astrobiology is the use of machine learning and artificial intelligence, AI, to analyze the massive datasets generated by telescopes, rovers, and laboratory simulations. AI can identify subtle patterns and anomalies potentially indicative of life that humans might overlook. For example, algorithms can sift through atmospheric spectra to detect unusual gas combinations or temporal variations that match predicted biosignatures. Similarly, AI can assist in analyzing geological samples by recognizing microfossil structures or chemical gradients. Such computational tools are increasingly important as missions return more complex data and as telescopes search across vast regions of space. Autonomous systems powered by AI will likely become vital for future probes exploring distant oceans, beneath icy crusts, or navigating rugged terrains on other planets, allowing for real-time decision-making and sample selection optimized for seeking life. Another key aspect of the search is planetary protection. The responsibility to avoid contaminating other worlds with Earth life or back-contaminating Earth with potential extraterrestrial organisms. This principle guides mission design, sterilization procedures, and sample handling protocols. Even the possibility of discovering life demands that planetary bodies remain as pristine as possible to preserve scientific integrity. In light of these efforts, it's worth noting that the absence of evidence is not evidence of absence. Just because we haven't yet found definite proof of alien life does not mean it is not out there. Our search methods are still in their infancy compared to the scale of the universe and the diversity of potential life-bearing conditions. What we know encourages us to keep looking, refining our tools, and expanding our understanding. In sum, the search for alien life depends on combining many approaches, from studying planetary atmospheres and surfaces to understanding life's origins and resilience. Each mission and scientific advance narrows the field of possibilities and informs where and how to look next. As exploration continues, the hope remains that we might one day answer one of humanity's most profound questions. Are we alone in the cosmos? As we advance our search, the role of upcoming and planned space missions becomes crucial. In the coming years, a suite of sophisticated spacecraft will be sent to explore new worlds with enhanced capacities for detecting signs of life. NASA's James Webb Space Telescope, JWST, launched recently, is already revolutionizing our view of distant exoplanets by analyzing their atmospheres with unprecedented sensitivity. It can identify chemical fingerprints of gases that, on Earth, are strongly linked to biological activity, such as oxygen, methane, and even water vapor. These observations are vital because they can reveal atmospheric imbalances that might hint at biological processes operating on exoplanets hundreds of light years away. For example, oxygen and methane coexisting in significant quantities may suggest an ongoing replenishment by life, as these gases would typically react and cancel each other out if present without biological input. JWST and future telescopes like the Extremely Large Telescope, ELT, and the 30-meter telescope, TMT, will provide even more detailed views, potentially pinpointing worlds with life-friendly chemistry. Closer to home, missions to the icy moons of Jupiter and Saturn, such as Europa Clipper and Dragonfly, hold exciting potential. Europa, for instance, is believed to harbor a global subsurface ocean beneath its ice crust, with possible hydrothermal activity on its seafloor that could sustain microbial ecosystems. Dragonfly will explore Titan, Saturn's largest moon, where a methane cycle resembling Earth's water cycle operates. Though Titan's chemistry is vastly different, it might host life forms based on alternative biochemistries, broadening our concept of habitability. Robotic missions to Mars continue to probe the planet's past and present conditions. The Perseverance rover, equipped with an array of instruments, is collecting rock and soil samples with the goal of returning them to Earth for detailed analysis. These samples may contain signs of ancient microbial life or chemical evidence of habitability that is impossible to confirm remotely. Detecting such biosignatures on Mars would be a watershed moment, reshaping our understanding of life's distribution in the solar system. Meanwhile, breakthroughs in cell biology and molecular analysis on Earth provide complementary insights. Studying extremophiles and synthetic biology experiments gives clues about possible alternative forms of life that could differ significantly from terrestrial examples. For instance, scientists have engineered bacteria capable of incorporating non-natural amino acids into their proteins or surviving with radically different genetic circuits, suggesting that life's chemistry might be more versatile than previously thought. This leads into a broader philosophical and scientific discussion. How do we define life? On Earth, life shares common features such as metabolism, reproduction, response to stimuli, and evolution by natural selection. However, alien life might not conform neatly to these criteria. Astrobiologists consider life as we don't know it by expanding definitions that include any system capable of information storage, replication, and energy utilization, even if these systems do not fit Earth-centric biology. To address this challenge, scientists develop frameworks to recognize biosignatures, which can range from molecules, isotopic ratios, and atmospheric gases to physical structures like microbial mats or distinct mineral alterations caused by biological activity. Importantly, these biosignatures must be distinguishable from purely abiotic processes, requiring careful interpretation and often complementary lines of evidence to rule out false positives. This cautionary approach was exemplified by the Viking landers' experiments on Mars in the 1970s. One test initially seemed to detect metabolic activity, but later analysis suggested that the results could be explained by chemical reactions in the soil rather than living organisms. Today, with advanced instruments and methodologies, scientists design experiments considering such ambiguities, improving our confidence in any potential discovery. The search for alien life also intersects with the study of potential technosignatures, indicators of advanced civilizations such as electromagnetic signals, megastructures, or pollutants in exoplanet atmospheres. The SETI Search for Extraterrestrial Intelligence initiative has used radio telescopes for decades, scanning specific star systems for unnatural signals. While no confirmed extraterrestrial transmissions have yet been found, this effort expands the search beyond microbial life, probing the possibility of intelligent, technology-producing civilizations. Detecting technosignatures presents its own challenges. Signals may be rare, short-lived, or use communication methods unfamiliar to us. Yet, rapid advances in radio astronomy, data processing, and machine learning enhance the possibility of identifying subtle or transient signals embedded within vast datasets. In addition, projects like Breakthrough Listen use powerful arrays to survey thousands of stars, multiple frequencies, and wide fields of view, widening the net for potential discoveries. As our instruments and strategies improve, so too does our understanding of how unique or common life might be. The field grapples with the Drake equation, a probabilistic formula estimating the number of civilizations in our galaxy capable of communication, balancing optimism with uncertainty. Key variables include the rate of star formation, the fraction of planets with habitable zones, the emergence of life, the development of intelligence, and the lifetime of civilizations. Each factor remains poorly constrained, but benefits from ongoing discoveries about exoplanets and astrobiology. Interestingly, searching for extraterrestrial life is not only an outward-looking endeavor. It also enriches our comprehension of life on Earth, its fragility, and its resilience. Studies prompted by astrobiology have revealed the planet's interconnected biogeochemical cycles, the adaptability of life forms in extreme and changing environments, and the delicate balance needed for complex ecosystems. This knowledge informs conservation and efforts to protect our biosphere amidst global change. The quest to find alien life also raises profound cultural and philosophical questions. What would it mean to discover life elsewhere? Would microbial organisms fundamentally change our place in the universe? Or would we await signals from intelligent beings to reshape human perspectives? Such reflections inspire outreach, education, and collaborative international projects, underscoring the unifying power of exploring questions that transcend national and disciplinary borders. Despite the enormity of the task, the continued development of technology, interdisciplinary research, and expanding explorations leverage humanity's collective curiosity and ingenuity. From analyzing the chemistry of distant planets to drilling into the ice of moons in our solar system, each step brings us closer to answering whether life exists beyond Earth. In summary, while definitive evidence of alien life has not yet been found, the trajectory of scientific progress is encouraging. We have identified promising environments, devised sophisticated detection methods, and launched missions that probe with increasing sensitivity. It remains a frontier where patience and perseverance are essential, but where the potential discovery would undoubtedly stand as one of the most transformative moments in human history. The search continues with a growing toolkit, better knowledge, and a persistent question that has driven explorers and thinkers for centuries. Are we alone?